Donc, bonsoir tout le monde. Je suis content de vous voir, de voir vos noms en moi. Je suis Jonathan Weissman, professeur de génétique, et je suis actuellement dans un colloque de retreat du LabX UMI, mais ils sont en post-session et je ne pouvais pas rater le Seminar Series. Donc, ça fait presque 2 ans qu'on a cette série gérée et coordonnée et animée par les étudiants de magistère. Le semaine Téléthon, c'est toujours un grand moment de magistère. Donc, il y aura des crêpes, il y aura plein de choses, on va vous dire. Et les relations entre BGA et le magistère et Téléthon, c'est une relation qui date des années. Donc, on est très content d'avoir Serge avec nous. Je vais laisser Félix présenter, mais n'oubliez pas de revenir. On a encore un séminaire avant Noël, donc le 7 décembre. Donc, n'hésitez pas de revenir pour le prochain. Félix, à toi. Et merci Serge d'être là avec nous.
Thank you, Jonathan. And so thank you everyone for being with us tonight. So today's edition is on the theme of the Téléthon. And I would like to remind that the Téléthon have a TV program on the 3rd of December, and that you can also donate even before that on the website don.téléthon.fr. Also, the BGA, so the student organization of the Magistère, is as a caritative pole, which is organizing events next week to collect funds for the Téléthon. And their social media will be in the chat also. And you can find them this week in the hall of the University Library, Grand Moulin. And so for tonight's edition, we are glad to welcome Dr. Serge Braun, who is since 2005 the scientific director of the AFM-Téléthon, so the French Association against myopathy, and is also the president of GENOSAFE. So he spent his whole career on the study of neuromuscular pathologies. He first did pharmaceutical studies and its PhD at Louis Pasteur University in Strasbourg. And then he moved to the USC Neuromuscular Center of Los Angeles for his postdoc. And after that, he came back to Strasbourg for 4 years. Then he left the public research to join for 10 years as research director the company Transgene, which is a leader in the field of gene therapy. And thereafter, he joined the AFM-Téléthon as a scientific director. And most recently, he became member of the French Académie Nationale of Pharmacy. And tonight, we are really happy to hear him talk to us about the research strategy and achievement of the AFM-Téléthon. So thanks again, Dr. Braun, for accepting the invitation, and the floor is yours.
Thank you, Felix. Actually, you should not thank me. I'm here to thank you, because you are all involved in a festive event for the sake of the Téléthon. And I'm here to explain briefly what has been made possible through following your involvement, and thanks to all the festive events for the country that happened during the Téléthon. So I'm gonna share my screen and start the presentation. Hope you see it well. So just as a reminder, the Téléthon was launched in 1987. The patient organization that got this idea to transfer the US Téléthon to France with the French touch, which is not just a TV show, but also with number of local events throughout the country. This is particular to France. And actually, the biggest Téléthon in the world is the French Téléthon. It was a small association before the Téléthon, founded in '58, but it became big thanks to the Téléthon. So it's a 30-hour TV show, 200,000 volunteers, more than 12,000 local events, the population over the years, the entire population over the years did participate to the Téléthon. And we are really thankful to the population because it helped us to raise more than 80 million euros annually. And most of it goes to research. This slide sums up our research strategy. As many other patient organizations, we do provide fundings to research labs, academic labs, but also private companies, biotechs mainly. Usually, we are funding about 250 projects every year, and also PhD and postdoctoral fellowships. Not only in France, 50% of our call for projects go to non-French labs, Europe, US, Japan, everywhere. And some of the projects reach the level of maturity that make them very close to the clinic, if not in clinical trials. And this means much bigger funding is necessary and long-term commitment. And so we, in this case, treat those projects differently. They are strategic to us, and they mean contract agreements with intellectual property aspects and return on investment as well. But there is always one clause that is non-negotiable, which is should the partner or the lab decide to stop the program, we have the possibility to get all the rights and pursue the program should we think it's worth it. And this is non-negotiable because we are dealing with public generosity. And so there is another aspect of our activity which is not very often encountered in the philanthropy sector. We are running our own labs as well. We have an institute of 700 people, 3 different entities. G.E.N.E, it's a non-for-profit biotech, 220 people dedicated to gene therapy. iStem, also in Évry, next to our headquarters. iStem is the largest French lab dealing with stem cells, embryonic and iPS stem cells. And the MEG Institute, which is in La Pitié-Salpêtrière Hospital, he's a 250 people institute dealing with muscle disorders, neuromuscular diseases, with INSERM lab, INSERM unit for basic science, patient care as well, patient consultation, but also a clinical investigation center for pediatric and adult clinical trials, and also teaching courses for neurologists, courses dedicated to muscle disorder. From this institute, some of the projects are transferred to startup biotechs. So spin-off of G.E.N.E Atamayo is a recent biotech dealing with gene therapy of limb-girdle muscular dystrophies, a subset of myopathies. Genosafe is a company dedicated to quality assurance and quality control of gene therapy products, and is acting for the pharma and the biotechs. Is a full private company now. We have also set together with BPI France, so the state of France, we set a venture fund dedicated to startup companies involved in innovative therapies for rare diseases. Because as you are going to see, we are not only involved in muscle disorders, but in all types of rare disorders. And you have the name of the startup companies that were funded by BPI France, And they are very successful companies. Some of them have now products on the market and have generated real breakthrough therapies. And I'll show you very briefly a few examples. And finally, we are now getting not close to the market, but we are reaching the market. And this means also partnership with pharmaceutical companies or with biotechs, the goal being to make sure that those innovations are made available for the patients. And the finance necessary to get a drug to the market is huge, is more than a Téléthon just for one drug. So we have to deal with private partners, and that's what we are doing in a number of programs. Originally, G.E.N.E was founded in 1990 to screen for genes. Originally, only a handful of genes responsible for rare disease were known, whereas more than 8,000 rare diseases are known, but the genes were not known. And G.E.N.E was built to set up a new technology based on robots to establish the human genome map, which was a quantum leap on the road of genome sequencing and gene discovery. In '92, the first human genome map was published by G.E.N.E and given free of any patent to the scientific community, and very symbolically here to James Watson, the Nobel Prize, who co-discovered the structure of the DNA, and who said that this human genome map will allow to gain more than 10 years in genetics. And actually, this turned out to be true. So we are not screening indefinitely for new genes. We are looking for therapies. And currently, more than 40 clinical trials are going on in rare diseases. I would say 50% for muscle disorders, 50% for other rare diseases affecting any types of organs. And 32 diseases are being targeted. This includes gene or cell therapy, but also conventional pharmacological molecules. And very importantly, some of the proof of concepts, as you are going to see, are applicable to frequent disorders. So the Téléthon is not only dealing with so-called rare disease, but is also providing new therapies and new technologies to all of us. And I would like also to maybe remind you that rare diseases are not so rare. If you consider the 8,000 rare diseases, this means 350 million people worldwide. So it's a real a public health issue. And this is the first example. And I'm talking about gene therapy here. The first proof of concept ever in the world of a successful gene therapy was provided by Alain Fischer, who I'm sure you know the name, and also teams in Italy, Maria Grazia Roncarolo here. And both of them have established a gene therapy approach that consists in retrieving bone marrow stem cells from the patient, put them in culture, transduce them with a virus that carries the therapeutic gene. Here it's a retrovirus. Now we are using HIV type lentiviral vectors. So the gene is integrates into the DNA of the target cells. Once the cells are genetically modified, they are injected by perfusion back to the patients. And the first proof of concept was shown in a so-called bubble boy disease. It's a genetic severe immunodeficiency, which is life-threatening. Most of the patients die during infancy. Their immune system is impaired due to their genetic disorder, and they cannot breathe the air. The air needs to be filtered, so they are confined into bubbles. One cannot touch them directly with our skin, which is full of microorganisms. And the only treatment is bone marrow graft, but bone marrow graft doesn't work always. And so there is no other options for those patients. And this gene therapy, after several weeks following the treatment, shows that the patients can build a functional immune system and start to live a normal life. We have now more than 20 years of follow-up. The patients are cured, and that's the term used by Alain Fischer now. They are cured. They don't need any treatment, and they have become adults and live a normal life. And this type of approach is also used for other diseases, other immunodeficiencies, blood disorders, including sickle cell disorder, for example, but also is now also tested in bone diseases and in adrenoleukodystrophy. This is a brain disorder, which, however, is due to a defect in bone marrow stem cells. You have here MRI pictures on the right of a patient at diagnosis, and 1 year later, 18 months later, 2 years later. And you see the progression of the disease with the degeneration, massive degeneration of the brain. And on the left panel, a patient at diagnosis just before gene therapy, and 12 months, 16 months, 2 years after therapy. And as you can see, the disease progression has been stopped by gene therapy. And this has become also a treatment approved by European and US drug agencies. The same technology, the same gene therapy is now used for cancer, the so-called CAR T-cell technology, which has nothing to do with Téléthon or the labs that have established this gene therapy approach for genetic disease. But the technology that has been validated in those rare diseases is now used to modify blood cells or bone marrow cells from cancer patients. The cells are armed against the tumor and injected back to the patient. So it's thanks to this gene therapy approach that was established in rare immunodeficiencies, industry is now treating cancer. The second wave of gene therapy that happened later on was the direct injection of gene therapy products into organs. And I am going to use here the example of a rare eye disorder called congenital Leber amaurosis. This disease is due to a defect in certain genes, such as RPE65. You can see the expression of the protein here in the retina of a healthy individual. Here you see the photoreceptors that are necessary to transform light into an electrical signal that is forwarded by the optic nerve to the brain. And without this layer of epithelial cells expressing the gene here in a patient, layer of photoreceptor is degenerating, and this leads to blindness before age of 20. There is a dog model of this disease. Briar dogs very often become blind. Those dogs have the same mutation as the human patients. Briar dogs were treated by gene therapy using a conventional approach to deliver the drug under the retina. And several weeks after this gene therapy, as you can see, the dogs recover vision. And this also has proven to be effective in humans. Here you have a young boy. He was treated in his left eye, but the eye is occluded by an obstacle, and so he cannot see from his left eye. And he's asked to walk along a way with arrows and obstacles. And since he cannot see, of course, he's unable to find his way. When the eye is freed of the label, the child is able to see from his left eye that was treated with this gene therapy, and he's able to find his way. This gene therapy is now an approved drug in Europe and in the US, and the same technology is now used to treat other blindness, genetic blindness. This is really spectacular. And now we are entering a third wave of gene therapy, which is the delivery of massive amounts of gene therapy product in the bloodstream to treat muscle disorders, to treat large tissues. For that purpose, I would like to take the example of spinal muscular atrophy. Spinal muscular atrophy is a genetic disease affecting the spinal cord, affecting motor neurons. The motor neurons are absolutely necessary for any movement. They transfer the electric signal to the muscle. The muscles are okay, but they cannot contract because of the loss of motor neurons in this disease. Patients affected with SMA, with spinal muscular atrophy, most of them die before 2 years of age. They are so weak, they cannot breathe by themselves. And of course, they won't be able to stand or walk. And the lifespan is really very short for more than 50% of the patients. Actually, 1 out of 50 people is a carrier. So among maybe someone in the audience is carrying this gene. Maybe I have the gene, I don't know. Judith Melki here discovered the defective gene named SMN in '95 with the help of the Téléthon. And Martin Barkatz, on the right picture, working at Geneton, set up the principle of gene therapy for this disease. She managed to find a virus having the property to, following IV administration, to cross the blood-brain barrier and transduce neurons, including motor neurons. And she showed in a mouse model of the disease that this gene therapy virtually cures these animals. The lifespan of SMA mice is really short, less than 3 weeks. And you have mice that have a normal lifespan following gene therapy. And this breakthrough was then transferred to the clinic a US biotech named Avexis was established and raised $500 million to initiate clinical trials in spinal muscular atrophy. And I'm going to show you now a video, a sound video, showing you the story of Evelyn. Evelyn is a young girl affected with spinal muscular atrophy, and her older daughter died from this disease. And her mother was pregnant before the diagnosis was made, and Evelyn turned out to be struck by spinal muscular atrophy. But Evelyn was able to, she was eligible for this gene therapy, and I would like to just watch and listen to this video.
Spinal muscular atrophy, or SMA type 1, is the leading genetic cause of death for infants today. A devastating fact this family knows all too well.
Our first daughter, she was diagnosed with SMA type 1 at 6 months of age, and she passed away at 15 months old.
This is a disease that is devastating for infants. A large percentage of patients die before 1 year of age, but 95+% are gone, unfortunately, by age 2.
SMA rapidly robs babies of their ability to move, talk, swallow, and eventually breathe. When Milan and his wife Elena found out they were pregnant with their second child, Evelyn, they prayed for a healthy baby. Unfortunately, that was not the case.
When she was born and the results came back and found out she was positive for SMA type 1, and we both kind of just broke down. We found out about the clinical trial here at Nationwide Children's Hospital.
This is really the first time that we've been able to apply gene therapy to any neuromuscular disease.
Today was a very special day for Evelyn. She was given genetic therapy for her missing SMN gene.
Babies with SMA are missing a gene vital for development. Through a one-time injection, gene therapy replaces this missing gene. For Evelyn, this gene therapy worked.
We start seeing changes as early as 2 months after treatment.
She started to push, and she started to get more active, holding her head up. And a little after 3-4 months, she rolled over on her own.
Now 3-year-old Evelyn challenges Dr. Mendel to a dance-off at her annual follow-up appointment.
She comes back after 3 years, and she runs up to me and hugs me and says, "Dr. Mendel, I love you."
I forget that she has SMA. This is a healthy girl, and she does everything that a normal 3-year-old child would do.
Something like that has never been achieved before.
With our first daughter, it was just devastating to lose a child. You lose all dreams you had for their future. And now we can actually save for college.
So this gene therapy is now an approved drug in Europe and in the US. And you can imagine the emotion of the teams at G.E.N.E that were involved in the initial discovery. Even though the first trial was held in the US, this is a breakthrough therapy that was born here in Évry. And now there are dozens of French patients who have been treated with this gene therapy, and 1,500 around the globe. This is another example, a muscle disorder named myotubular myopathy, I would say even more severe than spinal muscular atrophy. This disease strikes only boys. It's an X-linked genetic disorder. the boys are so weak, they can barely move, even their hands. And the gene was discovered in 97 in Strasbourg, and at G.E.N.E, Anna Bush-Bello, who used to work in the Strasbourg lab that discovered this gene, has established a gene therapy approach for myotubular myopathy. She decided to move to Ivry, join G.E.N.E. G.E.N.E having the capacities to produce large amounts of gene therapy vectors. She joined G.E.N.E and moved forward with this program up to the clinical stage. Through a partnership, through a collaboration with a US team that is holding dogs. There is a dog model of this disease, Labrador dogs, a colony that was found in Canada by a family, US family, the Fraser family, a former football player, US football player, with a child who died from myotubular myopathy. This family was aware of this dog colony in Canada and brought the dogs to the US. The collaboration started with Anna Bush-Bello, and you are going to see here what this gene therapy is able to do. This dog is affected. He has around 5 months lifespan. They are so weak, they cannot eat by themselves. They can barely walk. At this age, these young puppies should be much more active and should be playing with the veterinarians, but he's too weak. And without gene therapy, his life expectancy, as I said, is around 5 months at the most. So he's going to be, as you are going to see, he has been treated with gene therapy. He's a one-shot IV administration of an AAV8 vector carrying the myotubular gene. And we'll see the dog 2 weeks after injection. He can stand up, start to walk, shake his tail. And we are going to see the dog 2 weeks later, so 1 month after treatment. And he recovers. The dogs can reach adulthood and live a normal life of a Labrador dog. And this very spectacular gene therapy is now at the clinical stage. And we'll see during the next Téléthon, Jules. Jules is a French boy treated with this gene therapy. And he was treated in January 2020. He's making progress almost every day. He's now off of artificial respiration. He can stand on his feet. He can talk, and he's now going to school. And his parents and Jules himself will be brought to the TV, to the next Téléthon, just to show what gene therapy can do. And it's interesting because many of the breakthroughs in gene therapy were held in France. And this is an article by the Times in 2009 asking why is that France is so often seems to lead the way in gene therapy. And the explanation is very simple. It's because of the Téléthon. I have here the names of all the gene therapy drugs that are currently on the market in Europe or in the US. The first in 2015, and we are now 2021. And you have here the pictures of French researchers Alain Fischer, Philippe Lebouge, Martine Barkatz, José Alain Sahel at the Institute of the Vision Institute in Paris, Nathalie Cartier, Patrick Aubourg, who set the gene therapy for adrenoleukodystrophy. And also those drugs that are the CAR T-cell drugs, the gene therapy for cancer, inspired by the Bubble Boy gene therapy. So out of the 17 drugs that are now on the market, 12 owe to the Téléthon. And this curve sums up the history of innovation. And the gene therapy followed the same history. When the first idea comes up and the first proof of concept occurred, expectations were huge. And many people, including us, were convinced that medicine would be transformed by gene therapy. But it doesn't work always immediately. And sometimes side effects occur or failures occur. And these illusions become very quick and at very, very low level, to the same extent the initial expectations. But little by little, with progress, with improvements of the technology, with the real proof of concept, clinical proof of concept, the slope becomes to recover. And gene therapy found its place in, and is finding its place in the arsenal of therapeutics. And the same holds true for blood transfusion in the past, organ transplant, etc. And it took 30 years for gene therapy to happen. And without the long-term commitment, without the conviction of a patient organization like AFM-Téléthon, probably it would have taken even more than 30 years. And we all owe this to the Téléthon. This is the current status of GENETON. You remember the big lab initiated in 1990 initially for the human genome map, but now involved in gene therapy. There are 10 products arising from GENETON that are currently into the clinic, and 8 to reach the clinic in the coming year or two. And some of them are now products that are recognized in Europe and the US through a partnership with the US company for spinal muscular atrophy, the gene therapy that saved Evelyn. And this is the name of a product that has a temporary usage authorization in France for an eye disorder, and that is being developed by a French biotech company funded by AFM-Téléthon. We have issues yet to solve, and one of them, a crucial one, is large-scale production. We have established a GMP production facility for gene therapy vectors next to GENETON in Évry. The company is named Hyposkesi. Hyposkesi is producing gene therapy products for GENETON, but also for many other people around the globe. It's now a full private company, and is producing batches for clinical trials all around the world. The facility is now too small, and there is currently a new building which is under construction next to the initial one to produce more batches, because gene therapy is really popping up now. with a huge expansion. I would like also to take another example. This is a disease which is very symbolic of AFM-Téléthon. It's a Duchenne muscular dystrophy. It's also a muscle disorder that strikes boys, and with death occurring between 20 and 40 years of age. This is due to the absence of a protein named dystrophin. This is a normal muscle, and this is an abnormal muscle. The protein is absent, and the muscle is degenerating. And there are new technologies such as antisense oligonucleotides. Those are molecules that are able to modify RNA splicing, so RNA metabolism. And those antisense oligonucleotides have been developed for Duchenne dystrophy and also for spinal muscular atrophy. And it's a very interesting technology. It's a sort of synthetic RNA that is now approved in Europe and US for certain patients with Duchenne dystrophy, and also is being developed for other muscle disorders and potentially for all types of diseases. And actually, RNA treatment, we all heard about messenger RNA and the Moderna and Pfizer vaccines. And why did it happen so quickly? Not because huge amounts were put on the table and agencies were very keen to examine the dossier for a quick approval. No, it's because the technology was first studied for more than 20 years in rare diseases. And messenger RNA is not working very well in rare disease, but is working enough for vaccines. And if those vaccines came out so quickly, it's because 20 years of experience in rare diseases. And the first RNA treatments or drugs were first commercialized in Duchenne muscular dystrophy. A final example is with cell therapy. Here, the team at ISTEM in Évry, led by Christelle Monville here, was able to generate retina or retina cells from stem cells and use those patches to treat retinal disorders. You can see here this therapeutic patch delivered underneath the retina to treat retinitis pigmentosa. There's a clinical trial currently going on with very promising results. But interestingly, this approach would also be useful for macular degeneration, a very frequent disorder. ISTEM is also developing human skin from stem cells and is preparing a clinical trial for ulcerations of sickle cell patients, and is working with EUR G.E.N.E to generate new stems of this time not artificial stems, but this time with human skin, which could be useful for all types of skin conditions. And is also using robots to model genetic disease using iPS cells, iPS-derived cells from patients, and screen for molecules to correct the abnormalities that can be reproduced in vitro. And several drugs have been identified by ISTEM and are currently in clinical trial for different rare diseases, including autism. And there are other programs, registry programs that we are pursuing with a network of French labs specialized in mitochondrial disorders. Mitochondrial disorders are responsible for hundreds of diseases, of rare disease, but mitochondria are also involved in all types, actually virtually all diseases, including aging. And there are currently gene therapy product. One is about to reach the market for an eye disorder, but due to mitochondrial dysfunction. So there are gene therapy products that are able to allow protein production at the mitochondrial level. And this type of gene therapy could be also useful to treat mitochondria in other conditions, including non-genetic conditions. So the proof of concept may be transferred to a frequent disorder here as well. So I showed you through the different examples that innovation that are established for rare disease may be also useful for frequent disorders. And this network of mitochondria specialized labs is able to generate models in yeasts. So it's a screening for drugs on yeast model of genetic diseases. And some drugs have been identified that we are planning to test into patients. This program is named MitoDrug. And we are hopeful that those conventional molecules can also make a big difference in disease that for most of them currently have no treatment at all. I would like to finish with, you see those two tables. This is the total number of ongoing clinical trials in different diseases, muscle disorders, non-muscle disorders, gene therapy in green, cell therapy in blue, and pharmacological molecules in red. So a number of programs going on, and a lot of folks. And you have here the history of the research activity thanks to the Téléthon, from the first Téléthon in '87, the first proof of concept in rare immunodeficiencies, to now the revolution of bioproduction and of myology that we are preparing, following the revolution of gene therapy. And I would like to finish by this slide to give you rendezvous the 3rd and 4th of December, the next Téléthon, with more news about victories of gene therapy and other programs, a lot of hopes. And I would like to finish with a big thank you to you all, because through your event, the Téléthon week, you are collecting funds with crepe and balloons that are entering our labs. Drugs are coming out of those labs, and we owe you a lot. And I am really, really thankful to what you are doing, because without the Téléthon, nothing of what I have showed you would have happened. Thank you.
So thank you so much, Dr. Braun, for your great talk. So now it's time for questions. If you have questions, you can write them in the chat, and I will read them to Dr. Braun.
The first is always the most difficult.
Okay, we have one. So Camille is asking, What is the current price range for the single-dose drugs, and are those costs covered by the French health security for French patients?
That's a very important question, actually, because it's one of the big issues that we are trying to tackle. Some of the treatments are very cheap, but not gene therapy. Gene therapy is currently very expensive. For spinal muscular atrophy, the drug name is Zolgen SMA, Zolgensma. It's a one shot, but it's a €2 million worth shot. Very, very expensive, 2 million. And it's covered by the Social Security. The patient, the families don't have to pay for that. But why is it so expensive? There are several reasons. The main one, I would say, is production costs. If I take the example of conventional vaccine, a viral vaccine, for example, flu vaccine, what is injected intramuscularly in this type of vaccine is about 10 to the 7 viral particles. For Zolgensma, what is injected in the patients is 10 to the 15 to 10 to the 16 viral particles, 1 billion more viruses. And this means huge amounts of liters of bioreactors are necessary to meet the needs. Just for SMA, the estimation is around 10 million liters of bioreactors are necessary. So production is very, very expensive. So in order to circumvent this problem, we need to establish new production process, new technologies, disruptive technologies, and we are working on that. And it's very important because otherwise, this type of gene therapy won't be affordable worldwide. So it's important to first demonstrate that gene therapy is working, and little by little price will go down, like any other therapy actually. But it's a matter of fact that this type of gene therapy is currently very, very expensive.
Okay, thank you. We have another question. So Cassandra is asking, where is it? Is there a group funded by the Téléthon that's working on gene therapy for non-rare disease such as cancer?
Not for cancer, because they don't need money from the Téléthon. The pharmaceutical industry is putting billions into cancer, so there is no need for the Téléthon. But there are several frequent disorders that can be addressed through Téléthon fundings. I'll take one example. Several years ago, we were the first to initiate a clinical trial using stem cells in cardiac infarction. The heart muscle is unable to generate per se. There is no regeneration of the heart muscle. And the principle of this therapy is to inject into the affected area following infarction, is to inject stem cells that differentiate into cardiomyocyte to regenerate the affected area. And the first patient treated ever is a French patient in Paris. And we were interested in this program, not because it's our role to address this frequent disease, but we funded this work to help us to establish the proof of concept of this therapy that we would then transfer to cardiomyopathies that are frequently seen in certain muscle disorders.
Okay. There's no more question. I have last one question. Do you have program working on the malaria?
No, because it's very frequent disorder. It's interesting because, you know, it's a disease that there are patients who can resist to malaria. And those are the sickle cell patients. Sickle cell patients are very frequently found in populations in Africa, also in Asia, and around the Mediterranean Sea. And why is it so frequent? It's because those patients are less struck by malaria. They are resistant to malaria. And so there was, during evolution, sort of natural selection in favor of sickle cell patients in those countries. And so we are not working on malaria, but we are working on sickle cell.
Okay, thank you. And Jonathan is asking, is there any gene therapy to treat animals, or is it too expensive?
Well, it's first tested in animals, and hopefully in the future, some of those therapies will be made available for the animals, but you know, of course, not at such a price. So as long as prices are high, as long as production process are not tremendously improved, those gene therapy won't be made available for animals. There are some gene therapy already used in dogs, but mainly in the field of vaccines. So there are some vaccines that are actually gene therapy based, and they are currently used in dogs, but they are using technologies that are less expensive, plasmid technologies. This is much less expensive than viral based gene therapy, but plasmids are very poorly effective for our diseases currently.
Okay, so I think there's no more question. Thanks again, Dr. Braun, for your talk, and thanks the audience for being here tonight. The seminar has been recorded, so you will be able to find it on YouTube, on the YouTube channel and the website of the EUR G.E.N.E. Also, please come back to the next seminar, which is on the 7th of December. It will be with Dr. Duharcourt, and she will be speaking about epigenetic regulation in the paramecium. And also, don't forget the Téléthon event organized by the BGA next week. So wait, I have the list to remind you. So there is the stand in the BU at the university, in the hall of the BU. Next week there will be a stand where they will sell food in Lamarck building and in the Esplanade. And also you can buy tickets, tombola tickets. There is lots of prize to win, and so you have lots of chance to win, so you can do that. And yeah, I think that's it. So yeah, thanks again everyone, and thanks Dr. Braun, and everyone have a good evening.